This is Chapter 2, Lesson 8. Last time you were here with us, we covered PCR-3A Golden Gate. Today we'll be talking about gel electrophoresis. It sounds scary, but it's actually an extremely useful laboratory technique that allows us to separate mixtures of DNA, RNA, and proteins based on their molecular size and charge. Prior to using gel electrophoresis, we must obtain the desired DNA fragments. This can be done by using restriction enzymes. These enzymes act like scissors to cut the DNA into small pieces, giving us DNA fragments. Only with these DNA fragments can we finally pipette them into small wells in the agarose gel. We'll explain these terms more later. So how do you perform gel electrophoresis? Gel electrophoresis has three main components for the setup: the agarose gels, the sample walls, and the electrophoresis apparatus. The agarose gel is a porous gel used to separate DNA fragments. It is prepared using the included kit. The gel is made of a combination of 1 gram of agarose and 100 milliliters of TAE. Then the mixture should be microwaved until everything dissolves. The completed agarose gel should not be used before it has completely cooled down. Note that for the sake of this experiment, we will not be using DNA fragments, rather just red dye, so it's easier for you to see the lab. Here are the pipette tips, and this is the electrophoresis apparatus, a salt buffer solution that creates an electronegative current. So we take the agarose gel out and insert it in the chamber. Now we pour the TAE buffer, which ensures that the electric current flows through the gel. Next, we have to insert the DNA, or in our case red dye, into the sample well, but first we have to make sure that the pipette is set to 8 microliters. To perform electrophoresis, pipette the DNA fragment and place it into the DNA wells. This side is negatively charged. Insert the DNA fragments into the sample wells as shown above. Remember to use only one sample well for each DNA fragment. Given that the side with the wells is negatively charged and the other side is positively charged, the DNA, which naturally has a slight negative charge, will migrate from the negative towards the positively charged side. In terms of speed and length at which the pieces of DNA move, this entirely depends on the molecular weight. The molecular weight of the DNA strand is determined based on their lengths. The DNA strands that are smaller have less weight and will travel faster and further to the other side, whereas the larger, heavier molecules will not travel as far and will remain near the negatively charged wells. A DNA ladder is used to compare the size of your DNA with known sizes of DNA. This ladder helps us identify the number of bases the sample has. In the lab above, the dye seems to be relatively the same size in all the wells, as they all traveled similar distances at roughly the same speed. Well, let's take a look at this example. In this case, the DNA on the left is smaller or lighter than the DNA on the right, as it was able to travel a further distance, whereas the DNA on the right is much bigger as it didn't travel as far. So when do we use gel electrophoresis? In terms of gel electrophoresis application in the real world, it's used in forensic science to analyze DNA samples from crime scenes and compare those samples against the suspects. It can also be utilized to detect genetic mutations and variations associated with diseases such as sickle cell anemia and cystic fibrosis. Thank you for embarking upon this wild journey of electrophoresis.